Hello, I'm Eric Jorgensen from the University of Utah, and today we have Drs. Tom Reese and Jiang Heuser, and we're going to be interviewing them and discussing some of their seminal work that they did in the 1970s. They were using an electron microscope, which you see in the background. This is a device that uses electrons to image cells at a resolution close to 1 nanometer. So this is the highest resolution microscope possible. In the 1970s, they performed experiments that allowed us to understand neurotransmission, the fundamental behavior of a cell during a communication. And they're going to tell us about some of their experiments that they performed then. So what was the experiment? What was known, what was not known? And what was the experiment and what did it demonstrate?
We were interested in a little structure in the brain called a synapse. And this synapse, to me, is about the most important structure in the universe because it's the way that nerve cells communicate with each other. And it's these networks of communication that allow us to think and remember and be, be conscious and be who we are. And so the synapse is sort of the rock star of biology because of its tremendous interest and importance. And I've been interested in the synapse. There was a great deal already known about it. It was known about how it worked electrically. It was known that it released drugs. But nobody really had an intimate understanding of how an actual individual synapse works. And that's what we wanted.
Yeah, and I was equally interested and was graduating from medical school and made a beeline for the laboratory where the original work had been done that set the stage, where electrophysiologists had demonstrated that the communication event at the synapse involved the release of little packages of chemical transmitter in little electrical blips that they could see. Now, the man that did that, in whose lab I was joining, Sir Bernard Katz, went on to get the Nobel Prize after that, but his insight was profound, and on top of that, he'd written unbelievably beautiful papers that terribly impressed me as a student. So I made a beeline for that lab. He had already looked at electron micrographs and seen that the synapses on the inside were filled with tiny membrane vesicles, little spheres of membrane, and he made the brilliant but logical interpretation that those little spheres contained the packages of transmitter that he was measuring electrically as little blips. And so he had the insight to propose that the little electrical blips were being generated by chemicals coming from those tiny packages. That was established. I mean, it was such a clear-cut idea that it almost had to be that way. And so my project, when I entered that lab, and as a postdoc in those days, you're basically assigned a project, was to, quote, prove it. Get an image of these little vesicles popping on the surface of a nerve when I stimulate the nerve. And during that postdoc, I didn't get it. But I had the opportunity to join Tom Reese at the NIH. Immediately after that, we reformulated the problem and came up with a whole new approach to either proving or disproving that idea that Katz had had.
So Tom, maybe you could tell us how it came to be that you met John and how that led to a collaboration.
When John came to the lab, we decided to try a whole different approach. As John pointed out, usually you went to a lab as a postdoc and were assigned a project, but in his case, he came to the lab and assigned me a project, which was to work on this thing. So we were originally were thinking about looking at the aftermath and trying to interpret back to what might have happened, and that was a good approach.
As it turned out,
yes.
I mean, the idea, if these little packages are opening to discharge transmitter, and that's the mechanism of communication, then during that moment of opening, they should be able to capture material from outside the cell and carry it back in. And it would be an indirect proof that at least they had interacted with the surface.
So what you demonstrated was that the membrane, the synaptic vesicles were normally found in the synapse, in the bouton, and that they had visited the surface. And that's all you could show. And the problem was a question of time scale, or was there something missing?
there was the missing fact that we had nothing but embalming fluid, and we were dropping these things in embalming fluid every time, and life did not continue in any kind of appropriate manner in that solution, so we didn't capture it.
But the missing piece here, the next step at any rate, was when we decided, well, why don't we just stimulate the nerve for various periods of time and then fix it immediately, or let it sit for a while and have a look. And so we did a series of different amounts of stimulation and different time intervals for recovery, and that turned out to be very informative, because when you stimulated the nerve ending, where the vesicles were, got quite a bit bigger, as if the vesicles inside were being added to the surface. And so that was there's a second piece of evidence. And then if you wait a while after you stimulated before you fixed, you went back to a perfectly normal looking terminal, as if there was a cycle of activity and recovery. And that cycle, now we know that cycle involved the vesicle compartment. And I have to say, John, it's always been very clear to me that the idea of vesicle recycling was totally yours. Okay.
I wish.
What? No,
I
no,
wish.
really. I mean, I at the time, I was astonished by this idea, because it never occurred to me. And I'll take some credit later for some other steps. But
Okay. Well, it is true that the very first paper we wrote on the attempts in London to get it to work, where we didn't, but we got this aftermath effect, we said maybe the vesicles are dissolving and being reformed, or
you sort of had that in your head when
yeah,
you came.
and we said, or maybe it's merely recycling. Merely recycling is the final sentence of my paper in London. Merely, it became our career to pin that down and work it out.
So back in 1973, when you published this, was this accepted at that point? Was there anybody else who was doing these experiments at the time?
Oh, yeah.
Tell me a little bit about what was going on in 1973 when this paper came out.
73, there was a whole cadre of people doing this kind of work, all fascinated by the nervous system, all having electron microscopy as their tool. They've become experts at it. That was really the heyday of electron microscopy. It was the opportunity to see things for the first time at that resolution. So everything was a gold mine, and everything was fascinating and exciting, and people were going crazy. So there were a number of people that were neurobiologists who also focused on the nerve terminal. And the remarkable thing is that 2 or 3 groups, including groups here at the marine biology labs and in Europe, particularly in Milan, were focusing on the exact same preparation as us and doing the exact same experiments. And we were publishing in the exact same journals pictures that could have been swapped from one paper to the other. And isn't it in human nature? They were drawing completely different conclusions about how to interpret those micrographs. So there has never been peace in this field. There's never been consensus. And we are still, to this day, battling out the interpretation of what you see in an electron micrograph. But the way we were thinking was indicative of the way it's always been. Why we left everybody in the dust right then was because we're saying, how do you see it? How are we going to see that?
Yeah.
Well, we do this, we do this,
Yeah.
we put it on that substrate, we look at it with this instrument, that, etc. We'd moved instantly into the technology of it and getting the right image, period. But you can't forget that the rationale for doing it at all, for developing this machine, was basically to capture this event of neurotransmission. And the physiologists had taught us that the event occurs on 1,000th of a second. From when activity spreads into a nerve, and the nerve is going to communicate with the next one by secreting these little chemicals from synaptic vesicles. It takes only 1/1000th of a second for it to happen. And we needed to capture that event on that same time scale. We needed to capture it on its own territory. So that was why we went to all the effort to develop the freezing machine further.
So maybe, John, you could describe the instrument for our viewers and all of its glory and complications?
Well, I wish it were glorious and complicated, but it couldn't be more trivial. It's a copper block, copper chosen because it's a very highly conductive metal, the most highly conductive, particularly at those temperatures. So you have a heat sink, the best heat sink you can think of, imagine, and you want to apply the thing you want to study in the electron microscope, in our case, these little nerve terminals doing their secretory event, want to apply it to it, that prep to the copper block as abruptly and precisely timed as possible. So we just plain plunked it down on top of it, just popped it into it, touched it to the surface, to the copper surface at 4 degrees Kelvin. Had to hook up all the stimulating electrodes and develop methods of synchronizing that event.
And how was that done? How did you time when you would stimulate before it hit the copper block?
Simply had a magnet mounted right next to the sample, which could be used by a little sensor that senses magnets. As the magnet passed the sensor, it said, "You're about to hit the copper block," and that stimulated the electrode that fired the action potential in the nerve. And so we could synchronize that. Action potential arrived, the vesicles discharged, and the sample hit the copper block in 1,000th of a second.
But you also had a problem with the sample freezing before it ever hit the surface.
Oh, yeah, sure.
So it got so close to the copper that it pre-froze before it would get stimulated. But the real trick was, and still is, and you need to use it, the drug. The drug that augmented release. Our physiology friends who helped us work on it said either between 1,000 or 10,000 times, one nerve impulse would release 10,000 times more vesicles than normal. If you make a nerve release 10,000 times more than normal, it isn't going to want to do that very often. It does it once and it's shot. So we have to make sure we don't pre-stimulate it as we're getting the whole experiment ready.
So you do the experiment. What do you see? Was there a Eureka moment where you were on the scope and you said, "Oh my God, this is it. We seen the light. This is the image we were looking for"? I mean, can you describe a little bit about that? Were you together? Where did it happen?
As it happened, Tom wasn't there at the particular time that happened. He was back in NIH. It happened in San Francisco. But at that time I had, Mike Dennis was a very fine physiologist who was doing all of this treatment with this magical drug that increased things so much. And we had two young, even pre-docs, I think, maybe they had just gotten their postdoc, and they wanted to learn some EM, so they were wandering around my lab, sort of getting in the way underfoot. But one of them, Lily Jiang, and her husband, Jiang, who was the other person wandering around, both have gone on to great careers. But so they're sitting on the microscope, and they called me into the microscope, and they said, "John, I think we've got it. I mean, look at that." And I came in and looked over their shoulder, and I said, No, that's a capillary. You don't recognize there are a whole bunch of little vesicles attached to the surface of the capillary all the time. That's part of transport of blood. Nothing to do with nerves. You forget it. Looked a little harder. My God, it is a nerve terminal. Look at all those openings. And that really was a Eureka moment.
What was it you saw? What was it that you were actually looking for that was the Eureka?
Tom had explained that at that point we're doing it by freeze fracture. It's a method of looking at the surface membrane of the nerve by cracking open this frozen tissue. Particularly nice because, after all, we'd started by freezing it. So why not take advantage of that new technique? So we're cracking all these nerves open. It's a tube, so here's a long cylinder. And on that cylinder, dotting it all the way down, are these small craters, like craters on the moon. And it's a very small jump to
remember, the vesicles were originally lined up along this membrane.
Inside.
Inside, and you couldn't see it from the outside. You wouldn't have seen them at all. And so the fact that you could now you could see openings, it meant that they had actually exploded open under the surface of the just right before the muscle hit the copper block and frozen. So we'd captured them in a millisecond timescale, which is I don't think anybody really had done any anatomy structures of things on that time scale. There was another story that emerged right after that, and that is after the vesicle opened, we noticed that in the wall, in the membrane of the vesicle, there were various kinds of structures, and that as time passed, you see, with this freezing, you could do a time experiment. You could stimulate it at 10 milliseconds, 20 milliseconds, 30 milliseconds. So we noticed as time passed that the vesicle was getting wider and wider open, and that some of these particles inside, these structures inside, were coming out and diffusing into the surface membrane. And so that was we made a story out. We measured all that and made a story out of that. And so that was the third leg. That was actually, I enjoyed that part of it.
Well, it's a critical piece of the puzzle because if you have these little packages of transmitter and they're capable of fusing with the surface in a thousandth of a second, activated by calcium, what happens to them after that? And why were we seeing all these strange changes at much longer times? Well, that's when we really had fun with the freezing machine. We were no longer just trying to capture the immediate moment, but the aftermath of the event. As Tom described it, we can now do aftermath in really close coordination. We can be looking just milliseconds later. And it's our conviction that it's in the nature of a tiny membranous vesicle, something about biophysics of membranes, that when that joins a larger membrane like the surface of the nerve terminal, it has no alternative, but it has to collapse. It has to flatten out, join that membrane completely, and therefore deposit all of its contents in its own nature.
But did this end the debate? So you showed the vesicle touches, flattens out, and I mean, basically it's irreversible.
It's absolutely in the debate. Eric, you're baiting us because you know perfectly well that our colleagues in Italy, our colleagues at the MBL and so on, did not accept.
The siege of your times began at this point.
Yeah, they had to accept that Katz was fundamentally right, that that's how neurotransmission occurs, by the fusion of these vesicles. But then is when interpretation of electron micrographs and interpretation of how you did the experiment, because they looked at the same micrographs of a vesicle fused with the cell surface and concluded that that was it, that vesicle did that transiently. A little kissed the surface, let out transmitter, and simply reversed the process. Now, I was earlier describing to you that we thought it's in the biophysics of membranes that they can't do that, that if they've joined another bigger guy, they're going to merge with it completely. And so there is a controversy that is embedded in the literature and in scientific thinking to today. And everyone asks about this still. Are you really correct that all of your aftermath stuff is really true? Or should we believe the Italian group that it's just a kiss? The vesicles are kissing and coming right back in again in exact momentary exposure and then closure. And that's the true biophysics. And
so you then did experiments that were over longer and longer periods of time. And then you saw something that said, well, there is evidence that in fact these vesicles are collapsing fully into the membrane.
Yes, there are aftermath experiments of 30, 40, 50 milliseconds.
Particles inside the vesicle membrane.
Yes.
Diffusing out into the membrane.
The particles being our marker of membrane. Yeah. And now
the particles are places where the membrane is impinged by a transmembrane protein. And freeze fractures show up as bumps. And the bumps were diffusing out of the vesicle membrane into the whole other membrane. Now, it's not going to do that in a kiss and run.
What is it that you saw in your images that convinced you that vesicles had to be recycled?
Yeah, well, the reason I would have had a preliminary glimpse at that and said that maybe merely recycling happened was because of the magic clathrin-coated vesicles that appeared in the nerve terminal as a consequence of stimulation. And I had just come from my undergraduate work where I was exposed to the father of electron microscopy, Keith Porter. And he had trained so many outstanding people, but the person who was still present when I was in college was Tom Roth, and he had just discovered that the mechanism of endocytosis, the mechanism of uptake of material from the outside world in all cells, is mediated by a vesicle that has a coat, a beautiful cytoplasmic coat that actually is polygonal, like a Buckminster Fuller geodetic dome. And you can see that dome in the electron microscope. And so here I'm a young guy in London, and I see these domes in nerve terminals as a consequence of stimulation.
You'd seen those in London?
Yeah.
Oh, I didn't
And
know
I
that.
said, oh my God, it's endocytosis. And then it's also interesting that we managed to enter total contentious situation with our Italian competitors over that, because they couldn't see these coated vesicles. And why couldn't they see it and we did? Well, it was simply that technique of embalming we used. Ours was a little bit different than theirs. Ours had a little bit more heavy metal in it than theirs. So the coats looked a little more dramatic in our pictures than in theirs. But that's the way that electron microscopists can butt horns and can enter total disagreement, total contentiousness about an issue.
In the end, what I think solved the debate are those freeze etch images where you see the clathrin coat. This is absolutely clear. That geodesic structure is absolutely clear. Maybe you could tell us a little bit about the invention of the freeze etch technique and how you use that to show that there were clathrin coated vesicles.
That was a late chapter that arose from being able to directly freeze things, because if you freeze things out of in their natural juice, when you try to dry the water away, the contents of the juices collapse down top of everything. You can't see anything. So suddenly we were able to freeze, by being able to freeze directly, not even using fixatives, which also destroy things. We had a whole world of looking at the inside of membranes, and that was invented in our summer neurobiology course, and was told by one of John's students. Made a mistake on the machine, and John decided to have a look at it anyway, and that was one of those Eureka moments. All of a sudden, for the first time, we could actually look inside the cytoplasm, not just look at the membranes like you do with freeze fracture, but you could look inside the cytoplasm, and you could see these wonderful coats. And John went on back to San Francisco and began a decade of marvelous work looking at the coated vesicle and coated vesicle endocytosis, the coated vesicle cycle.
I think that's a good place to end it, but before we do, I would like to ask whether you guys have anything that reflections or memories or anything else you'd like to add.
Thank you for being my friend all these years. In spite of a few differences we've had now and then.
I can say exactly the same thing.
Yeah.
Thank you for being my friend and muse and colleague. And, it's just like, I don't know.